Uh, Assalamu alaikum everyone. I hope you all are doing well. So welcome in second.
Assalamu alaikum and a very warm welcome to everyone for joining us today. Assalamu alaikum and a very warm welcome to everyone. Thank you everyone for joining us today. Here is Hafsa Sarfraz. I am a student of MSc Honors Plant Breeding and Genetics from University of Agriculture, Faisalabad. Besides my educational career, I am serving as the General Secretary at Vita Nova International Alliance for Sciences. Now, I would like to call our today's instructor, Miss Hanfa. She is basically serving as a researcher at Vita Nova International Alliance for Sciences. Before we start our today's session, I would like to remind you all please present in the session, and if you have any queries, please ask at the end. You can write in the chat or tell me, we can give you a chance to become alive and ask your queries. Here is Miss Hanfa. Miss Hanfa, you can start now. The floor is yours. Thank you so much.
Okay, thank you so much. Assalamu alaikum everyone. Today we will discuss about the plus. We are about to explore one of the most essential tool in modern bioinformatics, that is BLAS, which is stand for Basic Local Alignment Search Tool, that is first developed in 1990 by Stephen and its colleagues. BLAST transform the way researchers compare DNA, RNA, protein sequences rather than performing time-consuming global alignment. BLAST use approaches meaning its local region of similarities quickly. and efficiently. When we talk about biological database like NCBI, gene bank, whether you are identify gene function, exploring evolutionary relationship or genetic mutation in diseases, BLAST give you a rapid and reliable insight. Today, I will guide you through its type and how we use NCBI web page. So, next slide. To give you a clear roadmap, here we have structured today's seminar. First, we will discuss about a quick introduction to do what BLAST is and why it's matter in biological research. Then we will explore the different types of BLAST like NBLAST, PBLAST, and when to use each. After that, I will walk you to through NCBI and how to input your sequence, choose databases, and set parameters. And finally, we will break down BLAST, interpret its result like e-value, percentage, identity, and query coverage. Next slide. What is BLAST? Bla- Now, let's define what is BLAST. BLAST actually is an algorithm designed to identify region of similarities between biological sequence. It can compare nucleotide sequence such as DNA, RNA, or protein sequence. Unlike global alignment tools that try to align the entire length of sequence, BLAST look for local high-scoring alignment. This approach is faster and biological meaningful, especially when identify conserved motif or functional domains. In research, BLAST is used to identify gene discovery, protein function prediction, phylogenetic analysis. For example, a scientist study a gene from a known virus can use BLAST to see what that gene has any similarity to human gene or to a gene in other organism that is very useful for our research. In its primary role in research to interfere functional and evolutionary relationship between the sequence and assist in gene discovery, protein functions. So let's move next slide. Why we use BLAST? So, why do scientists across the global rely on heavily on BLAST? First is it is effective for gene discovery, finding similar sequence across the massive database, and second, it allows us function prediction. If our unknown protein align well with other known sequence, it might be also actful as kinesis. Third, blast spot evolutionary studies that show how conserved a gene are across different organisms. And lastly, it's it's a vital disease research by comparing patient DNA with known disease related sequence. We can identify mutation that may contribute to condition like cancer or genetic disorder. Blast is the best part of the blast is that it is free and accessible through the NCBI website, making it to go to tools both for students and researchers. Next slide. Now let's talk about the type of blast. Now look at different type of blast. This is really important because using right blast program depend on what type of sequence we have and what we are trying to find. So let's start. Let's type first blast N. Blast N is nucleotide blast. It is used when you have nucleotide query and want to search it against a nucleotide database. And the second one is blast V. It is used for protein-protein comparison, widely used in functional annotation. Next, BLAST X. BLAST X is unique. It takes a nucleotide query, translate into all 6 possible protein reading frames, and search a protein database. T-BLAST N. It is reverse, like you put a protein and database is translated into nucleotide database. Then discuss about T-BLAST-X, which is more sensitive. It translates both query and databases into a protein. It is excellent for comparing divergent sequence through it. So if you have sequence like novel DNA region and suspect it to make or encode a protein, BLAST-X will help us to identify protein matches. If you have protein, non-protein, then want to find its gene and annotated genome, then use tBLAST N. Now in this slide we will discuss about the NCBI BLAST homepage. First, how to run a BLAST search on NCBI. When you go to BLAST NCBI database, you will see option to select the appropriate program like BLAST N, BLAST P, different type of BLAST, but commonly use BLAST N or BLAST P in our research. Then you will paste your sequence in FASTA format. That is a simple format where the first line is start with greater than symbol and followed by the sequence. For example, greater than then mark as gene and then all the code like A, T, G, C. Then you will choose your database such as no-reductant database or the organism specific one like human genome or mouse, anyone. You can also adjust the setting like E value, threshold, number of target sequence. Uh, when ready, then click blast within second. You will see the result will appear. Next, in this slide, you see that blast end for nucleotide sequence. Let's talk about the nucleotide sequence. Suppose you have a sequence or a new DNA fragment from soil microbe or any DNA from plant, animal, from marine, and want to find out what it's. Choose first of all the nucleotide blast, then paste your sequence in FASTA format, and then select the NR databases, and then optionally if you have to set E value and maximum number of target sequence, click blast. Within a short time, you will receive the match from similar sequences, possibly from bacteria, virus, or even any host, depending on your theory. BLAST N is perfect for gene annotation, species identification, or primary designing. Next slide. This presentation will be show that input query sequence like the left side screen that paste your nucleotide sequence in FASTA format in the query box on the NCBI BLAST homepage. Then select nucleotide blast or for blast N and choose the database and parameters. select after the parameter database, adjust the parameters, the e-value, threshold, each and everything set, and then click blast. In the next slide, you will see the interpretation result of blast n. Next slide. Here this shows the interpretation result in which discuss summary, graphical summary, description table, and accession number, e-value, percent identity, query coverage. So first of all, we will discuss about the graphical summary. Graphical summary show color bars. Red color for strong hits and blue color for weak, indicating which part of our query match sequence in the databases. And the next step is description table that lists the match sequence with key statistics. Accession number Accession number show that the ID of the sequence in the database. What is e-value? E-value, how likely the match is due to chance. Lower the e-value is better. E-value in BLAST is a basically statistical measure that tells us how likely it a sequence match occurred by a random chance than a biological similarities. The lower the e-value, the more significant the matches. It estimates the number of alignment with a score equal to better than observed one that could happen just by chance in a database of a given size. For example, e-value of 1 means you have expect one random match of similar quantity in the database. If we have the e-value of 1e20 means the chance of that match happening randomly is extremely low. That is suggests that a strong biological relationship. After that, we will discuss about Percentage identity that show how similar the sequence are. Basically, what is percentage identity? It is a key metric that tells us how similar your query sequence is to match sequence in a database. Specifically, how many positions in the alignment are exact matches. It is the percentage of identity-based DNA, RNA, or amino acid proteins between our query and the subject sequence. A higher percentage identity suggests that a closer evolutionary or functional relationship. Then queries coverage. Query coverage, how much of our sequence was matched. Query coverage tells us how much of our input sequence that like query DNA, RNA sequence was successfully aligned to a sequence in the database. It expressed as a percentage, but it helps us understand the match span, your entire sequence or a small portion. High query coverage like 95 to 100% means most of our all sequence was matched. Lower coverage like 20 to 30% means only a small fragment aligned, possibly a shared domain or a motif. Then we will discuss about alignment section. How the alignment section show the actual base by base comparison, highlights the matches, mismatches, and gaps. A stronger result typically show that low e-value, high identity, and good coverage. For example, we have 99% identity over 95% of our sequence within an e-value of 1e50 would be considered a very reliable sequence. Next slide. Exploring the protein with BLAST P. Let's switch to protein now. Suppose you have a amino acid sequence and want to know what protein it is. You would use BLAST P. First of all, you go to the home page of NCBI, click Protein BLAST, then paste your protein in FASTA format. Like greater than sign, FASTA format is used. Then choose the protein database. You can adjust the scoring matrix or gap if needed. When you get the result, you will again see a graphical summary, description table, alignment section, but here it is show that the amino acid identity, conservation, substitution, and non-protein domains. So if our query align well with other noun subject, they will high identity and conserve ATP binding domain. You can interfere it may be function similarly. Here is the more important three powerful uh, blast types, but that is less commonly used. That can be extremely useful in our research involving unknown genes, unannotated genome, or cross-species comparison. That are BLAST X, T BLAST N, T BLAST X. BLAST X. BLAST X DNA to protein, that is used for DNA to protein. BLAST X take a nucleotide, DNA or RNA sequence and translate into an all possible 6 protein reading frames. It then compared those translated protein sequence to a protein database. What is a reading frame? Reading frame is a way of a nucleotide sequence into a consecutive triplet, like we say codon. Each of which code for an amino acid since codes are read in a group of 3. The same sequence can be read in 3 different ways. We use BLASTX does not consume any withstand or frame contain the correct protein coding region. So it translates your nucleotide sequence in all 6 frames. Compare each translated protein into a protein database. Report match any from any frame that show significant similarities. It then compares those translated protein sequence to a protein database. It is useful as it helps us to identify potential protein product from unknown DNA, especially helpful when the function or even the coding region is not yet known. For example, you have a sequence, a novel gene from a coral or a plant or animal, whatever you require, but you don't know what it codes for. You can put that DNA sequence into BLASTX, and if the translated sequence match a noun protein, then we say that BLASTS will tell us. First of all, we go to the page of BLAST NCBI, click BLAST X, and paste our nucleotide sequence in FASTA format. Select a protein database or then click BLAST, and that result will show a graphical summary of alignment between translated query and noun protein, a description table listing protein hit with e-value, percent identity, and alignment score. A translated protein alignment show how your DNA map to protein sequence. Like this help us to identify possible coding region even if the DNA was not annotated. Then we will discuss about T blast N that is used for protein to translated nucleotides. Its mean reblast and take a protein sequence and search it against a nucleotide database. But it translates the database sequence into all six reading frames before the comparison. So, why it is useful? It is useful. This is ideal when your protein is known, but the DNA data we are comparing is not annotated. This is often used for the gene discovery in a new genome or transcriptomes. For example, we are discovered a new human transcriptome factor and want to see if it exists in newly sequenced fish genome, but the fish genome does not have known gene annotation. With the BLAST T and T-BLAST N, we can search our protein against the translated fish genome to find similar coding region. So, how to search T-BLAST N? First of all, we go to the NCBI, choose T-BLAST N, input our protein sequence in FASTA format, choose the nucleotide database like whole genome for transcriptome data, then submit our search. Then result will show the region in the translated nucleotide database that produce a strong protein alignment. Information on which DNA sequence code for similar proteins, possible location of an unannotated gene or open reading frames. T blast X. T blast X is translated nucleotide versus translated nucleotide. What does it mean? It's mean that this is most sensitive and sensitive blast variant. It take a nucleotide sequence first of all and translate it to all six frames and compare into a nucleotide database that is also translated into a protein into all six possible frames. It is useful as T-BLAST-X is the best comparison for divergent species. It is basically used for divergent species where the DNA sequence have changed significantly, but protein coding region will still conserve. This can reveal deep evolutionary relationship between homologous species even when the DNA sequence identity is low. For example, suppose you are comparing a plant transcriptome from an action fern to a flowering plant database. The DNA sequence may have diverged too much for BLAST-N to find a match, but using T-BLAST-X, which compare translated protein contents, may reveal the hidden homologies. First of all, we go to NCBI, then search, and then select the T-BLAST-X and paste our nucleotide sequence in the nucleotide in FASTA format, then choose a nucleotide database, then click BLAST. It takes some time. After that, result will show the protein level alignment between the translated form of both query and database. That hit like might be completely missed by BLAST N or BLAST X due to DNA divergence. It is especially valuable for the cross kingdom or gene studies. Basically, between blast X, T blast N, and T blast S, the main difference is query type. First of all, blast S is used query nucleotide, T blast N query type है protein, T blast X use the query type nucleotide and word and it will give the predict unknown protein products. Search for the gene in unannotated genome and T-BLAST-X is used for find distant homologous between the divergent species. We will discuss about a sequence, a novel DNA region from a species, from plant or animal. Then I want to know that what it does using BLAST-N because the DNA is so different from unknown sequence. But if I use BLAST-X, which is translate my DNA into all 6 reading frames and compare the resulting protein to a protein database like Swiss, SPOT, or NR. So here why we prefer protein level comparison BLAST-X, T-BLAST-N and T-BLAST-X when DNA sequence are too divergent. Because DNA evolved faster than protein sequence over a time, even related species. It may take very time different DNA sequence due to silent mutation or codon changes, but their protein sequence remain conserved because they carry out essential biological function. When we talk about BLAST N, fails to find the homology due to high divergence. Tools like BLAST X, T-BLAST X help us by translating the sequence or revealing hidden protein level similarities, giving us function evolutionary insights that give us the direct DNA comparison misses. So why we choose BLAST X over T BLAST N? Basically, we choose BLAST X over TNA because when our query is nucleotide sequence and it's code for a protein, but we are unsure that reading frame or function. For example, a novel gene in a marine organism, we use T-BLAST-N. When we have known protein sequence and want to find related genes in a nucleotide database, especially unannotated genome, for example, searching a human protein against a newly sequenced genome. Key takeaway and next step. To wrap up here are the most important point. BLAST is the most essential for biological sequence analysis. It is fast, accurate, and free. Choosing the correct variants like anti-blast, N-blast, X-blast, and T-blast and T-blast X is critical depending on our input and goals. Always check the e-value, percentage, identity, and query coverage to interpret result correctly. Explore additional features on the BLAST site like format options, organism filters, and graphical tools. And the most important, you should practice these all the BLAST types. Okay, thank you. Any question?
Thank you so much, Miss Hanfa, for the such informative session about the blast. And I think the questions that arised in previous session are clear now. And some questions are in the chat box. If you allow me, I can read the question and you can answer them. Miss Hanfa, are you there?
Yes, I'm
Okay.
audible.
Yes, you are audible.
Okay,
Okay, I am taking the questions from chat box and you can answer them.
okay, sure.
Here is a question, Mr. Mughal. Is the FASTA format standard? And I have heard about the FASTA queue. What is the main difference among them?
FASTA format that is basically used in BLAST because it is more accurate. We consider it is standard way to represent our DNA, RNA, protein sequence in bioinformatics. It is widely used in our simple and readable by both human and software. If there is simple sequence we put in the blast, it will not move to our further research.
Okay, thank you. Here is the next question from Fatma Zia. Can you please tell the difference between blast 10 and blast X at the...
I recently explained that blast and blast X. Like blast is basically local alignment. We use only the main genome of any query, while blast X our main type of blast X. Like blasts have different type blast and T blast V, blast X and T blast N and T blast X. While blast X, like we will discuss here that nucleotide our input like DNA or RNA sequence. And it will show the translation of our nucleotide sequence into a protein reading frames against any protein database. Then we suspect our DNA might be code for a protein, but we do not know that the exact protein. Then we use blast X. The DNA is unknown and annotated, but we want to see it. Then use the blast X.
Okay, here is a question from Muhammad Asif. Please describe the difference between global alignment and local alignment.
Okay. Like global alignment have whole genome sequence, while the blast is local alignment show like small genome or small part of our sequence, while the whole alignment show that the whole genome of our uh, species. Like if we global alignment in which we compared like two sequence across their entire length, like bacteria or any species from their beginning to end. But in blast we just use the FASTA format short sequence used to our research query.
Okay, thank you so much. It's a very good question. And now next question from Hamdik. Why do we need to compare between different DNA sequence? I mean, what applications could we benefit from after doing that?
Again question.
Why do we need to compare between different DNA sequences? Like he's asking about the applications of comparison between DNA sequences.
Okay, so comparing different biological sequence like DNA and protein sequence, like this question. Comparing
Yes.
our DNA or RNA or any protein sequence to identify gene in different organisms that evolved from the common ancestor, or comparing any gene from other species to see what they perform the same function. And it is basically used for the predict function of any species, unknown species, or comparison with unknown species sequence, or also with known species.
Okay, thank you so much for the explanation. Here is the next question. How can BLAST be used to identify potential horizontal gene transfer events between distantly related species? And what limitations might affect the accuracy of such analysis?
Okay, so first of all, we will discuss about BLAST related to sequence comparison. BLAST is directly used for our comparing sequence. It is widely used, like in bioinformatics. It takes any query sequence and compare the sequence. But here is the limitation of BLAST. BLAST is fast, but no guarantee to find the best possible alignment. It may miss weak similarities because it does not align every base or any amino acid. If for example, if we have two sequences are very distantly related, but BLAST might not detect them as similar. BLAST only perform for mainly matches only the most similar part. If we want whole sequence comparison, global alignment, we need other tool. It may not use the whole sequence comparison. It is used for only short sequences.
Okay, thank you so much, Miss, for the explanation. Here is the next question from Tahajwid. Why might you use T blast N instead of blast P?
P plus.
N instead of blast P.
Okay. While blast N is nucleotide blast, while P blast show that protein blast. Basically, we use if we have the sequence or any research related to a nucleotide sequence, we use blast N, while the P blast is used for the uh, protein query. But if we want to structure of protein, it may no work. It will move to fight to different other tools.
Okay, thank you so much, Miss. Here is the next question from Hans Raj. Why is it important to compare protein sequences instead of nucleotide sequences in some cases?
Protein sequences.
Instead of nucleotide sequences.
Okay, but I think so. I discussed earlier. Okay, I again told you that a nucleotide sequence is just made of building block of DNA or RNA. That is called nucleotide. DNA, adenine, thymine, and cytosine, guanine. While in protein sequence, we use the amino acid which are encoded by the nucleotide sequence, like transcription through transcription and translation.
Okay, here is the question from Tahajwed. He's asking if we are running blast and got multiple hits with very similar e values.
Yes, आवाज
तो
voice.
अभी क्लियर है?
Hello.
Miss, I am audible to you?
Yes, 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 yes.
Okay, here is the question from Taha. He is asking if we are running blast and got multiple values that are similar to closer to e value, then how we select the relevant one?
Like mostly use the lower e value, like 1e 50, 1e more than 50. That is much more reliable for our query.
Okay, here is another question. In data science, which type of blast we prefer to use? Blast X, Blast N, or T blast?
Again question. Blast.
Which type of blast we use in data science? Blast N, X, or T blast?
It is depend mostly use protein blast and nucleotide blast or X blast. T blast X and T blast N is rarely used. It is used for like unknown genome or divergent species. So basically, we use in our basic research as BS and MPhil who use nucleotide blast and protein blast.
Okay, thank you so much, Miss. I think all questions
Okay.
are done. If you guys have any other question, you can write in the chat box and we will address from Miss Hanfa. Yes, Mr. Mughal, FASTA format is the standard format we use in analysis here.
Already
Anyone?
discussed.
Yes,
Yes.
already discussed. I think
Mm.
he has missed the point. Yes, you can all write your names in the chat with your identity, like roll number, so we can take the note of attendance. And Miss Anfa, it's all done.
Okay. Yes, yes.
Okay. I think there is no more questions, so we are going to end our today's session. Thank you so much, everyone, for being here, and a huge thanks to Miss Hanfa for showing her presence today here. Thank you so much. Allah Hafiz.
Allah Hafiz.
Allah Hafiz, everyone. Thank you so much.